So you want to make a new species. Well, the first thing you're going to need to do is isolate that group of individuals you call a population from the existing species. Let's talk about that in today's lecture on reproductive isolation. I would like you to be able to list and define all species concepts. List in order the stages two populations go through in becoming two species. That's going to be both this lecture and the next. Give examples how reproductive isolation can happen very quickly in a single generation, or very slowly, or of course not at all. And describe and differentiate between different mechanisms of speciation. Let's get rolling. So, what is a species? Well, that's a good question. What a species is really up for debate. If I were to say there are this many species on the earth, I would immediately be met with a contradiction saying no, there aren't. Why? Well, because some are going extinct even now. Why? Well, because some people don't consider two things separate species. Why? Well, because the definition of species is a little fluid. So how many species are there? I don't know. A lot. So there's a lot of variety on Earth, and it really raises the question, why aren't there fewer species? Why does the Amazon need 58 different species of tree-dwelling frog? Does it need it? Well, define need, really. Does it have it? Well, it probably has more than that. And why are there that many when one niche could be taken up by just one species that is much more of a generalist instead of 58 different specialists? Are the two related populations separate species or not? Well, that's another good question. Are Highland cattle and Egyptian cattle the same species? Are they the same species as the American bison? Are they the same species as a guar? Are these all different species? And what is their relation to the ancient aurochs? Maybe they all could be one species. I know at least one man who considers all of those, including wildebeest, I believe, to be a single species. So why do we even care? I think it's a good question. Is there a moral or ethical value to a species? Does a species have a right to live? If we cause a species to go to extinction, is that actually an ecological problem? Or is that a moral problem? Or is it even, maybe it wasn't really a species at all? So there is a good number of reasons why we may or may not even care about each individual species. But first, we have to be able to define them. So what is a species? Well, we're going to use a certain thing called species concept. This is something that defines what is and what is not a distinct species. So we are going to be able to say this is and this is not. And it runs into the problem of what is a cryptic species. This is where two separate species are not identified as two separate species by a species concept. We have here 8 different species of bumblebee. And you might look at them and say, well, that's clearly 8 different species. Look, they're different sizes. I would like to remind you that in the early spring, bumblebees are larger when the queens are out, and then as the year goes on, the individuals actually get smaller. We have locally a bumblebee species you may be familiar with if you're spending time outside called Bombus vosnesensky. Now, Bombus vosnesensky is, there you see the queens in the spring. They're a bumblebee. They've got yellow face. The colloquial name is the yellow-faced bumblebee, and they're very common among all of our flowering plants. So they're the most common bumblebee species. We have others like Bombus sitkensis, Bombus melanopygus, and they really look different. But then you start looking even closer, and you have something called Bombus graciocollis, which looks very much like Bombus sitkensis, but lives in different habitats. So they are different species, and you can tell them apart. But I think it's like the banding pattern and by how the antennae bend or something like that. But then there's a real tricky one called Bombus caliginosus. Now, caliginous means like uncertain, so it is in itself a cryptic species. It looks just like Bombus vosnesensky. So it looks just like the yellow-faced bee, but Bombus caliginosus has different male genitals. So the male genitals look very different on Bombus caliginosus than they do on Bombus vosnesensky. This was actually a big problem for me. I made the statement that there are 19 different species of pollinator that visit Scotch broom. And someone said to me, what if you're undercounting because of cryptic species? And I said, well, that's a fair question. They said, well, look at Bombus caliginosus. Are you sure that one doesn't pollinate Scotch broom? And I pointed out that only females collect pollen. And Scotch broom only gives pollen. So I would never see a male bumblebee on a Scotch broom. and haven't. I mean, I know what the males look like for different species. I've never seen a male bumblebee on Scotch broom. So I could not distinguish female Bombus vosnesenski from female Bombus caliginosus because they look the same and everything about them is the same. But the male genitals are different, so I'd have to catch a male, but that male wasn't on Scotch broom, so you could never know. And my paper on this subject was actually put under intense scrutiny until I would call it Bombus voscal, as in Bombus vosnesenski / caliginosus, and just admit that I couldn't tell the difference between cryptic species. Because this really does matter to some people. So let's look into the different species concepts and look away from bumblebees. First is the morphospecies concept. And I gave that lengthy example because it really is just that confusing. And it really does matter just that much. So it's not just an aside, that's an example of this actually does matter. Now the morphospecies concept is careful discrimination of species based on phenotypes. And that's exactly what I was using in my bumblebee example, is I was using the morphospecies concept. Basically, if it looks like a duck, quacks like a duck, and acts like a duck, it's a duck. So the morphospecies concept is going to basically look at these organisms based on what we can observe. So Bombus sitchensis has this banding pattern on its body and has this type of smoky color on its wings. Boom. Bombus graciocolus has a slightly different banding pattern and a slightly different smoky pattern on the wings. That's a morphospecies concept. A problem with the morphospecies concept is that males and females look different. So, for example, of the bumblebees, I think it's Bombus melanopygus. The females have a black bottom. Melanopygus means black bottom. And they have a red band next to the black bottom, either one segment away or two segments away. The males are smaller and are totally yellow. So if you were just using the morphospecies concept, not only would you get these two mallards wrong, because they look very much like different ducks, but you might get the male and female species of Bombus melanopygus wrong. Then we have the phylogenetic species concept. And the phylogenetic species concept is going to cover species based on what we know about what they're related to. Basically, can you find a common monophyletic group? This is a very good one to do if you are using genetics for these. If you're taking a gene and sequencing it for all individuals and using a computer program to use the most parsimonious groups in the most parsimonious phylogenetic tree, and you find out that all your B's there all cluster together, your E's all cluster together, and your G's all cluster together. Now, I'm not saying what they look like. For all you know, they can all look the exact same. But if there are three groups of ducks and they share a common ancestor and traits within that group, well, then that's a species. So we're defining it as a common ancestor. So then the G's and E's are definitely not the same species because there would be a polyphyletic group. The G's are more related to F's and the E's are more related to C's and D's. So this is not a monophyletic group, the E's and G's, but they are within themselves the same. And that's a phylogenetic species concept. And for sometimes when you're using just genetic sequencing of a large number of samples, people go out and they trawl the ocean for DNA, and then they just take the DNA, sequence it, and they can make these kind of trees without ever really seeing the organism. Which, from my perspective as an ecologist, is boring. But from some people's perspective as a geneticist, is the way. So I've actually dabbled in both, and really pluses and minuses for each. This is what we generally use in biology, is the biological species concept. The biological species concept is if that group is reproductively isolated from other species. And if this duck mates with other ducks and produces viable, fertile baby ducks, then it's a duck. So they are going to look at whether things are going to be reproducing with one another. What I'd be doing here is I would say that Bombus caliginosus is going to be distinct from Bombus vosnesenski because even though we can't tell the difference, they can. And since they can tell the difference, a caliginosus would never mate with a vosnesenski. And even to them, they look completely different and act completely different. And they are genetically isolated, and therefore they are biologically isolated. Now, the biological species concept tends towards making the phylogenetic species concept true, because reproductive isolation is the first step in speciation, after which you get reproductive isolation, then the accumulation of mutations, and then they can't mate with each other after a time, which enhances this. So they can't produce viable hybrids after a time. Now, we're going to be going through this with several lectures, of course. Here are some examples where that kind of breaks down. We have here the Solanum species. So Solanum lycopersicans, schmielewski, and pimpinellifolium, Cheesemanii, sorry. So, lycopersicans, Cheesemanii, and pimpinellifolium, they are all interfertile. You can cross any one of those any way to any one of the other ones, and you're going to get a viable, fertile offspring. Now, Solanum pimpinellifolium is a sprawling vine that has very small tomatoes. Cheesemanii is a sprawling vine that has medium-sized tomatoes, and lycopersicans is your common tomato, which is not so much a sprawling vine. It's got some determinate growth to it. But what we have here is is very different species that are in morphological forms. You could tell the difference between these three if you were to look at them in a field. But they can't tell the difference as far as breeding, so they can interbreed perfectly. And then you have Solanum parviflorum. And we see it's parviflorum, right? Peruvianum, sorry. Solanum peruvianum. And you can see how one group of Solanum peruvianum is more related to Solanum schmielewski, and the other Solanum peruvianum is more related to Solanum chilense. So these are different species, but they look the same and they interbreed with one another. It's the north and south varieties of peruvianum found in north and south Peru that are more or less related to schmielewski or chilense. The south is related to chilense, and you think your South American geography, Peru and Chile are close together. Schmielewski is growing up in the northern area. So that would be a problem because we would consider it one species and they are completely interbreedable with one another. But if you look at it from the the phylogenetic concept, they are different species. And then you have Solanum pinellii. And that's really where the nightmares begin. Because Solanum pinellii can reproduce with Solanum hybrochides, which is right next to it, but only if Solanum pinellii stigma accepts pollen from habrochides, not the other way around. So it's a unidirectional reproduction. What we actually start seeing along this whole Solanum genus, oh no, not the whole genus, but the whole tomato complex, is that you're going to see if you take pollen from one and put it on the stigma of another, you can get them to reproduce sometimes but not others, and the back and forth is actually far more complicated, leading to a really messy group. So, the biological species concept looks good, but starts to break down. And there is a problem there. And that is exactly what you would expect if you have a group of populations that have been undergoing isolation. They have accumulated mutations, but they have not yet become such that you can't interbreed them. And this is another core problem with the biological species concept. In nature, they don't reproduce. Well, then why am I saying they can reproduce? Because if you take them to the lab, you can mate them a whole bunch of different ways. In fact, you put them under enough stress, you can do a whole bunch of things to them. So this is a core problem of the biological species concept, is what things do in captivity is very different than what things do in nature. These dogs, we don't know what they would do if we were to actually just let them run free. Would a pit bull mate with a German shepherd? Probably. Would a bulldog mate with a German shepherd? Probably not. Would a chihuahua mate with a German shepherd? No. Any self-respecting German shepherd will just eat the chihuahua and be done with it, okay? We give it one shake, two shakes, squeak toy, done. So that's a nature versus forcing thing. Can you take chihuahua sperm and artificially inseminate a German shepherd? Yes, you can. I mean, you kind of feel bad for the German shepherd at that moment, but you can do it that way. And there are groups who hold that the biological species concept is true only in what could hypothetically breed. And this is actually a core concept of a different species concept we're not talking about here. They believe that false killer whales, and dolphins are the same species. And that's the same species as porpoise. Why? Well, because if you put them in captivity, you can get what's called a wholphin. So a wholphin is a dolphin-whale hybrid. Now, it wouldn't be viable in the wild. Anything that attempts mating and could hypothetically mate is considered a species by that group, which is also going to take bison and cows and say, well, they can mate, and you can get beefalo, so there's some viability to that. The biological species concept can be used in an extreme version called baraminology, anything that could hypothetically mate. And this is where you have to take a real step back, because you are thinking viable fertile offspring or not. If you think viable fertile offspring, then you start drawing a lot more lines there. But if you start thinking, well, they could attempt to mate, then humans and chimpanzees are the same species, because humans attempt to mate with chimps occasionally. And dolphins will attempt to mate with anything. Dolphins are very sexually active, and dolphins will attempt to mate with humans, and some humans do attempt to mate with dolphins. So that same species would consider humans and dolphins the same species were it not tied to religious practices that believe that would be bestiality, because humans are clearly a different species. Based on what species concept? Oh crud, theirs falls apart too. So basically what I'm showing here with some very odd examples is that any species concept can be torn down given enough scrutiny. Yes, there are problems with the concepts in addition to mating. Bacteria and archaea are going to share DNA. When I say share DNA, I mean they just pick up DNA from the ground and just put it in themselves. So that kind of horizontal gene transfer runs havoc with the phylogenetic species concept. Morphological, you can't tell the difference between bacteria. No, you can't. No, you can't. If you say you can, well then look at the different ones. I'm sure there are plenty that you can't tell the difference between. The biological species concept, well, what's mating when you are asexual? Yeah, different mating systems make isolation more or less difficult too. So that's really there are a lot of problems with the concepts. And I spent more time saying what problems there are than what the actual concepts are, because I want to get you the idea that evolution is messy. Evolution is messy. So let's look at an application to this, because we actually do apply this. So let's take the European common skate. Now you're going to look in your book and it's going to show that there is a little map for where the European common skate lives. Now morphologically you can tell the difference between the two of them. They have different types of dentition on the outside of their skin. They have these kind of teeth on their skin, and they look very different. So you can tell the difference based on some morphological traits. Phylogenetically, you can see here from this phylogeny that they're not a monophyletic group. There's that long-nosed skate that kind of intercalates between the flipper skate and the blue skate. So there are different types of species going on there. Biological, in nature, they do not meet and mate. So given any one of these species concepts, you would put them as two different species. Now check your book to see the actual outcomes of why that would matter to the fishing industry and such. But this is also one of those instances where we would see the species concepts may break down if you take two skates and put them in a cage together. No kids. They will. And they may produce viable fertile offspring, or at least they'll try. So that is one place where the concept may break down a bit. All right, speciation itself. Speciation itself has to undergo several different things. The first of which, so you're going to have isolation, divergence, and secondary contact. There's your list. And that's when they may or may not actually reinforce that initial reproductive isolation. So first off is that isolation. Here we have two types of camels. The Bactrian and the dromedary. Dromedary looks like a D, it's got the single hump. Bactrian looks like a B, it's got the two humps. There you go, never get them wrong again. The Bactrian camel can be found in the Gobi Desert of Mongolia. The dromedary camel can be found in the Sahara Desert and the Arabian deserts. So these are isolated from one another. They don't mate, not in nature, because they're never going to meet each other in nature. So you have isolation. You can see a clear divergence in the number of humps. And what do you call a camel with 3 humps? Pregnant. Okay, so these are isolated. There has been a reduction in gene flow between the two populations. So this can be done in a couple different ways. There's allopatric speciation, is what we're going to talk about first. It's speciation that occurs in 2 separate spaces. And this can occur by something called dispersal, where you have a core population. Let's say the camelids initially existed in, wait for it, America. Yes. So the camelids initially existed in North America. Now they actually dispersed down to South America, where we get our llamas and alpacas and guanacos. They also dispersed twice, probably, to Arabia and the Gobi. So this is 2 dispersal events, and the dispersal population was then isolated from the source population. So the dispersal here is going to mean a new population forms through something like a founder effect and is genetically isolated from others. Then we have something called vicariance. We're going to split two populations from one another. So a vicariance event may have occurred. Let's just hypothetically say that the camelids dispersed into Asia, and what happened there is at some point the Himalayan mountains began to rise too far for camels to actually cross between the Gobi and the Arabian peninsulas, and just the area between just got too harsh for them to live in. And that would be a vicariance event where you split the population into two subgroups. So here's an example of a geographic isolation by dispersal. So this is geographic isolation here of different Drosophila species, fruit flies. What happened is there have been repeated founder effects. You started here on Kauai, and then they have been founding the new islands as the new islands came out of the sea. So as Hawaii, our new big island, came out of the sea, it was colonized by Drosophila, which are now genetically isolated from the other Drosophila. So this kind of genetic isolation by a founder effect followed by a population explosion on a new area can cause that divergence. And that is a genetic drift thing as well, where we have the founder effect is going to mean genes are going to go to fixation quicker in that small population, which is going to enhance the probability that they're going to be genetically distinct from and unable to breed with the source population. Another example is vicariance. Again, your book does a very good job here. We're actually looking at the rising of Panama. It separated the Caribbean from the Pacific. So when the Caribbean was separated from the Pacific, you see at first these shrimp that lived at the greatest depths, they couldn't get over this mid-ocean ridge. And these couldn't get over this mid-ocean ridge, so they were then separated. I don't remember if that's A, B, C, D, or B, or F, but it's one of those populations. Then the ones that lived in mid-waters became separated by the ocean ridge. And the length of time since they diverged is actually correlated to how deep they live from each other. So this is one of those really good records that as Panama raised, so these species were isolated from each other in real time and became different species. So these shallow water species, I think that's B, show the least divergence, B and D I guess, maybe even A. They show the least genetic divergence from one another, but they were all different species to begin with. So Caribbean E and Caribbean A are different species from one another, just like they're different species. The E group and the A group are different species from one another, just like the Pacific and Caribbean versions are different species from one another now. Then we have something called sympatric speciation, which is when you're going to get speciation. These are allopatric. And these again, this is sympatric. That would be polyploidy. What's happening here is that there's a mutation causing the production of diploid gametes. Self-fertilization occurs, and now you have a 4N individual. This 4N individual now produces 2N gametes. These 2N gametes can't go back to reproduce with a 1N gamete because they'd make a triploid organism, which is not reproductively viable. So essentially, within a single mutation and single duplication event, you've created a new species. It's going to look just like the adult. We don't know how often this happens because we don't sequence the genome of every single organism we encounter. We do know it happens a lot in plants. This is a common thing in plants. And that's going to make a new species, which over time may actually look very different from the parent species. What I want you to think of is, as well, that 4N species is now, it's tetraploid. It has 4 copies of each gene. It has 4 copies of each gene. It can accumulate 3 different versions of 3 different alleles and still have a dominant allele running them all. So the amount of mutation that can happen within that is huge. And the probability of speciation goes up. So we do see a lot of genome duplication in plants. It's not that common in animals, though. So the more you know, right? But you're going to see a whole bunch of ancient duplication events when you look through the genomes of plants. Polyploidy and sympatric speciation through polyploidy is pretty common in the plant kingdom. Then you have mutation, where a single mutation can actually make for a new species. Another type of sympatric speciation. And what happens here is a single gene can make a new species. So We have left-handed and right-handed snails. It's a single gene that can mutate and makes these left-handed snails unable to reproduce with the right-handed snails. The problem is this happens in multiple different populations. So this can go back and forth, and a species can basically switch between one from another basically by a single mutation. Single mutation makes a completely new species because they are reproductively isolated from others of the previously same species. So if it's really for any sexually selected trait that's same as the preference, like this, the sexual selection is to be able to mate, and they'll circle each other for a while, and then they get bored and go away. This can lead to very rapid speciation events. So we do see speciation occur in real time. The last thing I wanted to cover is just kind of give you a preview and a preview taste is the accumulation. The accumulation of mutations. The divergence. If you have two populations that cannot reproduce with one another, each one of those populations is going to genetically drift on its own. And because they're going to genetically drift on its own, you're going to get substitutions, and you're going to get fixation, and you're going to get the extinction of different alleles. And when this happens, we're going to end up with two different species that look very different from one another because that accumulation of mutations is going to make them morphologically distinct, phylogenetically different, and biologically incompatible. So check those three species concept I just mentioned and see if you can go back to them. Can new species rise in a single generation? Look at some of the examples I've given. And can two populations be isolated for millions of years and not become two species? Think it over. So in the next lecture, we're going to discuss other examples of speciation. And I urge you to read your books. Your book does a very good job of describing this and has some very nice examples.